Welcome back, everyone. So Lorena, you can share the screen. We can start with the second part.
Okay, so welcome back to the Braves that they are still there. So we are going to make a little bit of hands-on of some of the things that we have seen before. So we will be focused on mainly the mining of the genomes and the websites that we can use for those kind of analysis. Okay. So, well, one of the first things that I think is interesting to analyze is to how to submit or genomic information to NCBI. Okay, here you can find at the NCBI how once you have analyzed your genomes, verify, purify, and when you have something that is ready to be analyzed, as we say, one of the things that we have interest is to have the annotation of this genome. And one of the ways that we have talked about to do that is just to make the submission, to make it available in public databases. But the gene bank allow us to keep it safe for a period of time. For example, we have to submit our genome sequencing and make a restriction of 2 years for the time that we have going to analyze this data and publish it before this is publicly available. So one we can use these different systems to submit the different kind of information. So you have here the submission to the gene bank directly, and we have different ways to do that. We have we can prepare and using a command line program. Here is explained how to do that, but we can also do it in a very simple way through the website that anyone can carry on. So if we click here in the Bankit, we are going to go directly to the submit new sequencing to GenBank. Okay. If we want to do that, at some point we are going to register ourselves. Okay, because it's you need to as we say, you are going to submit the information, but you can restrict the access of this information, and this is a way that is keep it always to your personal access to um, this this data that you have included. So one of the first things will be to log in yourself and to create your account in which you can submit all the data to the GenBank. And when we have this, we can select all the types of information that we can include in this service, and one of them is going to be, for example, prokaryotic genomes. Okay, yeah, there is a question. Partial genomes, yeah, I mean, it means draft genomes, what we can deposit, because most of them today are drafts. But the idea is to submit as complete as we can. So once we have make all the analysis that we can do within our sequences, it will be the time to submit that information, not before. Um, And at that point, we are going to select this section for prokaryotic genomes. And when we make a start, they are going to tell us that we need to log in. Okay. So, well, when you log in here, for example, I can log in myself. Okay, you can use it different methods for login. I think it's interesting to use your ORCID because then all your scientific information is linked. Um, okay, so I'm going to connect to my one, but you can do directly once you have registered in the website. So as I have submitted several times different kind of information, now you can see that they have many of the data that they have submitted before. So when you get to your one, you can go to my submissions here, and then you can start a new submission. If you click here, okay, I think I have tried it from before. Uh, but this one probably. Okay, so once you click in a genome submission, it's pretty easy because they are going to ask you for all the main information that you should include. So you are going to decide which kind of information you want to upload. So you will be able to upload just the assembly, or you can even include one annotated genome if you have annotated by yourself using command lines, or if you have annotated in RAS, for example, and you want to update this kind of annotation, you can do it directly. So, but you just need to go one by one. For example, you can use update a single genome, but you can also use several genomes and update it at the same time if they are, for example, all belongs to the same kind of project or they are all related. You can do it at once. Okay. We are not going to make the whole process. It's just to show you that it is pretty easy. Once we have here is just selecting, for example, if we have a purified genome, we are going a single genome, we can click it, go for the next one, and then we are going to have, this kind of steps that allow us in a very simple way to make a whole submission. So we are going to have all data, the data of the people that are related to the generation of the genome, the people who is going to be included in the publication or whatever to want to include as a submitter, okay, with your institutions, and but it's mainly following and including the data. Then we will pass to the general information of the organism. Then we will follow for the files. So we just select these files that we want to include. In this step, we can also include the assembly, but we can also include other data that we have from our genome. We are going to allow its an assignment, include reference. For example, if we have already written a publication and we are going to just include this data for deliberation of the data that to be analyzed by the reviewer, we can also include this information already there. And the final step is going to allow us to revise all the information that we have included and submit that information. So it's going to be pretty easy to follow step by step once we are here and complete all this information. Okay. So in addition to that one, it's going out. Sorry, I need to find again where we have been. We're going to keep it smaller because we are going to be to the websites and it's going to be easy probably in that way. If you've seen before, when we could select the different options, we have as well one that allow us for sequence read archive, the SRR documents to be uploaded. This is what I told you before, that are the reads that you go primary from the company of sequencing. And this is the raw data, the real reads that were obtained from the company. It could include the adapters, or they could be already deleted, depending on who you are working with. This is an automatic step on their platforms. But in any case, this is the more direct information that we can get from sequencing, and this is one that I think is important, that will be submitted. This is even easier than the one that we say for the genome. Um, than for the assembled genome. We are going to go directly to um, sequence with archive. Okay. And here is very easy to, with the new submission. Okay, I have already one that I was testing yesterday to see how it was. Is even easier the information that we need to include. Again, we can include general information about the people who is going to submit the data, the SRI metadata. They are just included the files because, for example, for the assembling, we need to include a little bit more information about the technology that we have used, if this is Lumina, the kind of machines. You need to keep this information at the beginning, but for the sequences with archive is pretty straightforward to include all the data that they are required and submitted. So yeah, I just highly recommend it to do that. Just go to the NCBI and get those information. When you use it for deposit your genomic assembly, you can get finally this NCBI annotated genome, and you can use those data that directly for example, if you are interested in some specific genes, it's interesting to have those numbers that are assigned by this kind of annotation, because if you are going to publish through NCBI, this is the information that everyone can look it faster. So if you use this code in the nomenclature, it's going to be easy for the rest of the people to find. So there is the question. It is necessary to unlock the server file. We want to unlock the original. Yeah. What I say is that it's better to upload both. Because the assembly genome is the we have curated the data as we've seen before, and this data could be not um, we have curated them with the technology that we have at this moment. And if we only upload the assembly genome, we have already made many restrictions to the data. This is not the whole information that we have generated with the sequencing. What I mean is that if this is quite pretty easy steps, really it takes few minutes to complete a sequence with archive submission. I think for the scientific community, it is going to be much more better to have those information. Me as a reviewer, for example, is something that I used to ask to the people to include those data, because it could be very helpful later. So the idea is to submit for one side the assembled genome, and for the other side these original files from the read. Okay, so from the NCBI, I think is everything I want um, to show you, just how to submit your data that you need to create. Um, One of the programs that I want to show you as well is just so how easy it is to work with many of the websites of systems to analyze all data. So here you have some information. If you remember, we have said that this is one of the available websites to verify the completeness of our assembly. Okay, it's similar to the check analysis. You here have all the information, how it works, what they really do. If you are interested to go further for any of these. But if you no, wait a minute. So uh, you need to include a genome sequence. So one of the other things that we are going to do before moving on this is just to go to NCBI to download a um, a genome. Okay. So if we go directly to the website, we open the NCBI. I don't know if any of you have done this before, but just in the way that we can get some genomic information in the way that you can test directly all these programs. And if you have your own sequence or your own assembly, you can test it directly. But if not, we are going to just download some of them in this place. Just okay, no, there's no. Uh, so, for example, imagine I'm going to be interested in look for some specific genome of uh, micro monospora uh, Zika. Okay, so I look for this information. Ah, no, I include my say. Okay. So imagine that I want to download any of these genomes for my analysis. I have same that I'm looking for Zika. Well, in any case, we want to get Globispora, for example. Okay, so I going to be interested. I open the assembly of these organisms. Here we can find some of the information that we have told before, the genome size, the number of contigs. Look, this is quite a huge number of contigs for this analysis. But this is quite old. We have normally improved those data in the new with the new technologies. We have the N50, the L50 numbers, and all the information about the different methods. If you see, we have a download here, and we have also another downstairs. So we can go to the NCBI to download. And for the things that we are going to do today, we will need to download two kind of files. From one side, the general FASTA file, which is the one that we have worked until now. But for some of the analysis that we would like to do, we are going to download the protein sequence. Okay, because for the analysis of the carbon source utilization, we will need this. So we can just select those files. Here I can indicate that it's going to be the genome of Micromonospora to know which one is the one that I'm downloading. Okay, and it's going to start to be downloaded in a file. We can go directly to in our downloading section, and you see that this is going to be compressed. So we need to decompress. Uh, yes, it depends of the program that you have to do that to get the information of the inside these elements. So we have several documents, and inside the folder NCBI dataset, we can get into see the data, and we are going to have these two files. We get into one of them, and we can see the first one. We have the genomic information of the organism with the code that NCBI is using, and also we have a protein file that we have asked it for. Okay, so as this genome it has been analyzed by the annotation system of the NCBI, we have all these files available. So we have selected both of them uh, to be used in many of the programs that we have. We are going to ensure that that our extension is FASTA instead of FNA to ensure that in your system you can modify this extension, or you can download in a different way to get the FASTA. Okay, but carry on as the FNA and FASTA format, they are quite similar, and we can change it directly. I have questions related to genome-based identification. Let me know one by one. How do you see the MICA, TICS, and SDA clause? Yeah, really well the use of all of them. I use most of this system, so I think it works quite well. The microbial genome atlas is still limited for some compounds, not for all of them, but for some they are limited the information that we can get to compare, but in the other way is quite useful. Tix is the one that we have saw you before, and we will see also now how to use it. And SBO Cloud Stack, it works also quite well. So for me, all of them, they are useful for this kind of analysis. I believe these platforms they are much updated and recent type strains genome. Yeah, that's true. Do you think the identification of tyners from these platforms can be used straight away to claim novel taxa? We need to be very careful in this sense, um, just because sometimes you have you don't have the genomic information for several taxa. And this is something that we have to keep in mind, because many times we just compare with this kind of system that directly select the genomic information available and make analysis. And we forgot that our strains could be very close to another one that for which the genome has not been sequenced yet. So we need to keep maintaining doing the 16S analysis, the 16S phylogenetic tree, and check if it's one of the closest in this analysis, its genome has not been sequenced. In this case, we will be more sure that we are working with all the information, and we have taken into consideration all the strains that should be considered before to decide that we have a new species or not. In my case, I have genome sequencing of a Staphylococcus strain and identifying using MIGA and TIGS. Surprisingly, the TIGS reveals the strain is a potential new species. Okay, and the MIGA says that the strain belongs to the species Staphylococcus Rhizophila at the yeah, but you know you have a very low Ani value. So that's mean that it's not that species neither. You are, if you remember, we were talking about the value. Okay. I wonder among these two reports, we I consider clear information, the genome one. Okay. Okay. So, but you have a very low Ani value. So I don't understand why the your system say that it belong to this species because this is the closest species, but this not belong to this species with this Ani value. So you need to have a higher Ani value to be considered the same species. So for me, both results are in agreement. So also ticks and Ani value agrees that your strains that not belong to the same species of the closest sorry, Stenotrophomonas. Uh, how can we generate the protein file if we haven't submitted yet whole genome sequence file of my bacteria? Okay, this is an example of the one that is already there. We can do that using, we need to have annotation of our genome to be able to generate a protein file. So the first thing that we need to do is to have annotated of the genome. It doesn't matter which system we have used it for annotation. We can generate this file. So if we have annotated and we are not yet submitted to NCBI, we can do it also by command line to generate this kind of file. But if, for example, if you are analyzing your genome in RAS and you have submitted to the database, we are also able to download this protein file. Okay, you have different methods to get also this information. But you need to have annotated first your genome. Okay, so, well, here I continue with the files. So here we have both the FASTA file for the genome and the protein. And these are the ones that we can use in further analysis. So if we go back to the Gvolant that we have said that we can send information directly for analysis, you need to take care for the kind of files that we have submitted for each of the programs because not of them ask for the same kind of files. And this is important to check each time that we are going to use a different website or system. In this case, many kind of files, even compressed or uncompressed, can be used. But for example, if we have the multifasta file that we have now generated, you see, well, it goes directly, but if we can go file by file, we can go to the place we have download our data and decompress it, select it the same place that we have seen before and just go, in this case, for example, for the FASTA file, okay, select it. And once we have selected the file, just say upload the file. This is going to be uploaded, okay, and then they are going to ask us different information that we should include. For example, imagine we want to say that this is the micromonospora analysis. Okay, my email to be sure that I got the information back. Yeah, for if you want to analyze of the sequence, you need to keep this one number Okay, then we need to select the type of sequencing. In this case, we have said that we have used the FASTA file with the nucleotide. So we select this one, the genome. We can choose the different version of Busco or even TECMA to analyze or orthologs. So we select the latest version, the Busco B5. And then we should clarify here which kind of information we need to analyze. So to select the ortholog set. So if we to select this one, we have a whole list that if we click here, we see the whole list of orthologs that has been classified. For example, in this case, my microorganism is an actinobacteria and can select this one, just copy and just very limited sorry, I don't know why. Okay, so this is just another window that is open. I can close it when it's finished. Yes, I copy the information that I need and just select it here and okay, mark yes. The ortholog set that I want to use is the one that's corresponding to the phylum Actinobacteria. So once we do that, just say that we want to start of analysis and it go to another page um, for generate those data. I will be back here because I already done some of this analysis before, just to not wait for the results. As we have included our email address, we are going to be able to get our results there. And we are going to receive an email one is ready, and we can just a link to go directly to those results. And we will see something like this, depending on the data that we have included. And we will see the grade of completeness. Remember that we have said that over 95% is correct. So here we have 98, over 98. And then we have also information for the kind of genes that they are missing, because, and we can also check the information of the total number of genes that has been evaluated with this selected orthologs group that we have used. For example, for this Actinobacteria phylum, we see that they are going to be checked 292 genes, and we were able to find 288 genes. Okay. We are going to have other information about our genome, about the total length of the genome, this M50, this L50, the number of bases of the total composition of our genome, and also the GC percentage count. Okay, so if I click here, we can go directly to those results to see in the directly in the web. I don't know if there's any question. Okay, it's open. Okay. So we have seen here the real results that we got. We have the results, our database, and all this information. We can go further for all the orthologs that we found, the ones that we found not. Okay, we can see all this information and all the what I told you before about the number of genes, the percentage, how it has been working, the material, and all the information also about the program that we have used. So with this simple method that you have seen is quite fast. It takes some minutes to get some less than 1 hour normally to get an answer from this in your email, and you are going to be sure of the quality and completeness of original. So other analysis that I think is interesting and we can do now is, for example, to evaluate the contamination. We have checked um, the completeness of the genome sequence, but it is also interesting to understand which is if we have any contamination. So in FBO Cloud, you can also have this count S16S that, as I told you, we are going to have um, they are going to look for the 16s that are found within our genomic information in the way that if there are several ones, they will be able to determine if they are contaminated. So if we are going to, we can select between bacteria and archaea. Sorry, I'm going to give you the link in the chat as well. If I'm be able to discover this. Okay. Well, here you find the website and that's going here to the contest. Okay, here what we are going to do is just the same as we said before. Select bacteria. In this case, we are going to upload a pasta file. That is that we are going to select the file that we said before. Pasta file. And allow the system to upload it, and then once it's completely charged, we are going to run a contest. You see that directly once it's uploaded, we are going to have information about this genome, some basic information about it, and then once the run contest is finished, it's going to tell us if we have contamination or not. This is don't use to take too long, so probably we can wait until it's down. Okay, you see here, okay, it say that this project has only one 16S identified within the whole genome, so it cannot be checked for possible contamination. And in this way, we also get the 16S sequence that it has been analyzed. So to show you some examples of genomes with problems of contamination, here in FBO Cloud they have selected and they have analyzed directly information from GenBank that has this kind of problems. So if we click here into browse, we can see which are the results that we get for some of the analysis when several has been detected. For example, these Bacillus cereus, we can see that there are 3 fragments that there is no line between them, so they are very different ones. In this, it's supposed to be a Bacillus strain, but one of the 16s that is found corresponding to Saccharomyces, and there is another one that also corresponds to Bacillus, but it looks very different from the previous one. So we have the similarity between them, and this allow us to know that this should be some important grade of contamination because we can detect three different 16S from three different bacteria within our data. If we get a contamination, the only thing that we can do is sequence again or microorganisms because that means that the DNA was contaminated at the very beginning. So there is no way to be sure that some part of the information that we have got really correspond to our microorganisms. So this sequencing should be discarded and we need to start again from the beginning. Okay, so with this system, we can, I'm going to start to close because if not, we are going to have too many things open. Okay, and we can move forward to the next one. Is everything clear for these ones? If you have any other question, please let me know. And we can move to the following that I going, okay, I'm going to include the next one is the gap mine that we've seen before. Okay, here is one of the we open it and it's also quite simple to be used and this is very useful to determine the small carbon source utilization using our genomic information. For this one is for the one that we will need to use a protein file. So we are going to select again our file, but this time we are going to select this one, the protein FAA file, okay, from our genome. And this is the one that allow us to this program to interpret it if it has all the genes necessary to determine that it could use or not a compound. So when you upload directly, you will see it's going to give you some information about the process that it is quite fast. You see, and they give you that it has been take 11 seconds to get your result. So you see that it is something pretty easy. We can go to the view results and we have this kind of table. So we have here a lot of information. The pathway is the one that we have on the left and is determined if or based on the genomic information of our organisms, we can say that they are able to use these compounds or not. Okay, so you have a whole series of carbohydrates and amino acids and the determination. You can see that there are different colors. You can see that is remarked the different steps necessary for working properly the utilization of these compounds, and they have also the same colors. The colors, if you go to the bottom, you have the legend, and the legend indicate the ones that appear in green means that you have high confidence that with the genomic information analyzed, your microorganism is able to use those compounds. You have medium confidence when you have some of the genes that are reliable, but other ones are not so much. So you get a medium confidence. And when one of the genes is not detected and not the whole pathway is available, you are going to get a red that indicates this low confidence. So normally when we have low confidence is that we are not we have not many possibilities that our microorganisms use those compounds. But we can use this also for analysis in general. For example, we can see here that we have been doing the acetate, that this is not a standard compound that will use it, or cellulose, ethanol. So fumarate as well. We can design some specific media to make, for example, a specific isolation of this kind of organisms. Or we can also compare and go for a better characterization to test some of these in vitro to verify its capacity and its agreement with the genomic information. Okay, so I think it's something quite fast to obtain, and we can get an interesting information as well. Okay, here I could put some examples. Okay, for the following one, we are going to see how to submit information to the anti-SMAS. Um, do here. Okay. As we've seen before, anti-SMAS is also pretty easy to be used. Um, we have all this information about bacteria, fungi, and plants if you work in different things, and also we can um, include the information in a pretty simple way. You can include your email address. You can do both. Upload directly the file that you have, or if you have the NCBN number that this microorganism that you have already deposited in the databases, you can work directly with this affection number. And then you have several options that you have to decide the way you want to work. For example, normally is by default we are going to have a relaxed in the detection strictness, but we can be loose or we can be strict. What that means is that how the values that the program is going to select to decide if something could be considered as part of a biocluster or not. Then if we are going to be strict, we are going to be more sure of the information we can get. If we decide to be loose, we need to be more careful about those data and its interpretation. Okay, by therefore, we can let it relax it, the medium option one, and then we can select the kind, we have a default analysis that is proposed by the system, but we can also include other platforms or the other comparison to compare all data. What we need to think is that if we include many things, as much things as we include, is going to require more computational time. So it's going to take long to get our results. Okay. But in a general, we can keep this by default option and just submit our system. I'm not going to submit one now because it takes a little bit longer, so we are not going to be able to get our results just now. But I'm going to get into one of my last analysis in the way that we can work directly over the website with that one. Uh, you see, I already got our analysis for Volante, so it does not take too long to get the original ones. And I'm going to look for anti-SMAS analysis. You see, when you submit, you get an email like this telling you about the working job that you have submitted that has been finished and your information is now ready to be analyzed. So if I click it here, we can see all this information that I showed you before. Uh, I like the compact view. I don't know why, but it's probably because it was the first one that we see before in the system. And here we have all the regions in which a biocluster has been detected. We have the second column indicate us the type of biocluster that we have got in the main groups that we could be interested in. Many of them you see in this case are PKs, terpenes, um, NRPS. So we are going to have all this information. Another important point that we should check it is where is located this biocluster. We have the starting position and the final position where all of our genes are located. And why I say this is important? Because if we don't have a whole sequence, what happens is that we have contigs. And sometimes uh, when we have contigs, some of these biocluster could be in the middle of two of them. So when we have values at the very beginning of the contig, like here from 1 to 3000, or here, we need to be more careful that it is a real one or it is a partial one. Okay, that we need to evaluate very well the genes. And if we don't have all of them, it could be that it's just divided. So sometimes we got a number that is higher than the real number of bioclusters that we have. Okay, we should always keep in mind that when we generate a draft genomes, we can have this kind of problem. Well, with each of them, here you have also the information, and you can go one by one, get into the information and check for the different kind of genes, which are core genes. But normally here we have the terpene, that is the one more responsible for the production of the compounds that we are going to be interested in. And then we have the position of the other biosynthetic genes that we are going to be indicated in this color, the regulatory genes that we have found in green, and we can analyze all this information. Okay, so you can just go through the system and try different things to evaluate uh, what you got. You can have also, for example, uh, the your sequencing and compare with others available, or you can also get more information about each of these things. Okay, right. We can go. Well, many of you probably have used the six tool. I'm going to go also try fast to this one. Just give you as well the link to get into. Okay. Again, it's pretty easy. As you have seen, for most of them, you just need to include the assembly file and get most of the results. In this one, you have many information about how it works. The frequently question is quite useful as well. You have examples, etc. If you want to submit your own sequencing, you have to select this section, submit your query, and this is what we got. Then we are going to select our file, as we have done before with the FASTA file. And then, as you can see here, you can add or the gene bank number for closely related strain, or In the case you are not sure or it's the first time that you want to test, you have not to include, this is not compulsory. You can select to include other genome to sequences or not, as well as you can to restrict your analysis in the way that you only have these three with this selected strain or not, and you want to allow the system to detect the closest one and include it. So if we just include our sequencing and just go and include or email, just submit our query, and we are going to get directly generally the results obtained from the system. So if we do that, I'm going to look for one of my last results. Well, here, yeah, when we got the results, we got again an email directly from the system that allow us to go directly to the observe or result. And here we have a lot of information. We have, as I say, we can check the figure for the stickiness. We need to envelope normally for to be able to see the tree. You see here we have our query that appears here, and we have the closest strings that has been selected automatically by the system. Okay, in this case, we can verify that it looks like that it's different. It's marked by the system with a different species and subspecies, so it's different based on the 16F phylogeny. We can check also for the whole genome analysis. Similarly, we are going to have the phylogenetic tree with all the information. We can copy this image. We can make some improvements of what we can get there. We can download, but I recommended you not use all this image. We can download here in this position the network file for this image, and then you can use another programs to improve the imaging that you want to prepare for your publication, for example. So then we are going to have many other information about, as I say before, for the digital DNA hybridization, different values that will help us to analyze the differences with the closest related species. And we got another file with different general information about all the genes that has been included in the analysis, with the size of the genome, the percentage of the GC, and the difference in the percentage. That this is also an important one. In the cases, this server analysis data will be public or will be maybe produced alone. In this one, in the type genome server is not public. The information that we log here is just for us, but we don't need to create an account. We just got the information directly to our genome. Okay, so they are going to send us the information directly to us. Well. You have many other things that you can check one by one that will take time just to go finishing, because everyone should be tired. Just two things. One I think is important is the protologger. I want to give you the access as well in the chat, because mainly for taxonomists, I think, well, for everyone, but for taxonomists, it gives most of the information that we need and that we can analyze for a genome. So I think it's quite interesting website. Yeah, that you should know. Okay, it's going to be developed in through Galaxy. So, okay, and so this is the general one. You need to log in here in this one, and you need to create your account, and that is the way that you will be able to update data, submit data, and manage your own data. I thought that okay. I will have this. I'm going to open my one in a way that you see a little bit more information. Okay, so in this one is quite good that you have the explanation in the general one that I submitted to you. You have the explanation of the system, what is all the information that you get, how it works, and the reference, and also this quite useful instruction of how to use Protolog. So you can just check here how to create your account, introduce your data, and analyze it. Just to show you very quickly, once you have created your account, you can introduce here your data, you can upload your data and generate this tell Protologger to execute, yeah, or analyze those data. And then we are going to, once this has been finished, what we got is a list like this with all the analysis that has been carried out, and we can check one by one. So for example, we have the Protolog overview. If we check here in the display section, we are going to have many information about our genome that is classified in different groups. For example, here we have also the number of carbon suits that are produced to be used, including a whole list of compounds. We are going to have antibiotic target alterations, in the antibiotic resistant analysis. In this case, there are not. You have found also a list of enzymes that are being detected to be produced by this genome according to the genomic information. So this is also quite interesting, and we didn't see this for the other programs that we have checked before. And then we are going to have also an ecological analysis checking through max how many times our microorganism has been detected in other environments. So this is quite useful information. We are going to have a genomic tree, a 16S tree, and any values, and also KEF profiles or different analysis that could be done. So I invite you to play with all these options within your genome. I think you can get quite interesting results for your publication. Okay, so the last one that I want to just very, very fast talk about is the RAS system. So we can go directly, we can just go to RAS. If we click on Google directly, we can go to the RAS server. I can also include this address here. Okay, and we just need as I told you before, here we need to register. So when we arrive here, we need, sorry, it's going to be ask you for your data, login data, as we'll see here. You normally get this, so you introduce your data and then you can access to the RAS. So you need to solicit a registration. This is free, but you need to ask for it. and then you will have your own data available here. So here you can upload a new job to be annotated. Remember that we have said that this is one of the systems that we can use to annotate our genomes and indeed maintain this information private. So it's pretty easy, as we said before. We can select a file, a FASTA file, then go to the next step. I don't know. Mhm. Let me see. Well, it's just updating the information, the system. Well, okay. You see, it was reading the information of the genome, so we have this basic information again about the genome that we are trying to include in our database. And then it asks for some information about the taxonomy. We know the genus, the species, and here we need to make a mark in the bacteria or the microorganisms that we are using, and then go to step 3, just indicated that we are agree with all the data and submit to be analyzed. I'm not going to be submitted this one because it's one that is already carried out, but once we submit the information, it's going to take some time to do the analysis, and then we can go to the jobs overview here in this set. And here you can see some of the genomes that I have annotated through this system. And we can select one of them, for example, and go to see to view the details. We are going to have some information about the process for annotation, that if it has worked properly, we are going to have a checkpoint in each of the steps, and this allow us to have all the information available. Once this has been done, we can go to this directly through this link to the seed viewer. And this will allow us to have a lot of information about the genome and the genes implicated. I would say we imagine that we are interested in the secondary metabolism of these organisms. I can go through these, or we can find, for example, plant hormones. They are going to detect that we have 8 genes related with biosynthesis of auxins. We can get into this information, and we are going to be able all the genes that has been identified for our microorganisms, okay, which are detected if we go on each of them. For example, the tryptophan synthase. So I can go into the gene and get more specific information about this gene. I can go to the sequence that correspond to that gene. I can see the position within the genome, so I'm going to be able to understand which genes are upstreams or downstreams in my genome, and this sometimes is very useful depending on the information that we want to get. Just, well, there are many options here to compare, to look in your genome. You can use a sequence of using blast search to directly go here and introduce the sequences that you want to analyze if it's present or not in your genome. You can also use the genome browser and just to look for a specific name. For example, if we want to see which kind of ribosomes close, if we are looking, for example, for the 16S or any specific gene, the GRAs or whatever, we can go directly and look at the ones that has been annotated in that sense. It's pretty useful this section that I remember you that we can find it here in the genome book. So I think it's all the things that I want to talk to you in a general way for today. So if you have any questions, if not, we have finished. Just you can write to me also if you have specific questions later when you try to do any of this analysis.
Thank you, Lorena. You've been tremendously energetic in talking about the analysis of genomes and microbiome, and the students are still, rather not the students, but the participants are still there. You
Yeah,
know,
I'm quite surprised of that. I thought I have lost it most of them in the
Oh, no,
in
no,
the
no.
meantime.
Our workshops have been very popular. Even the last one, we had the same feedback. Today as well, we have the same feedback. I guess the participants have asked most of the questions during the session, so that's why there are no more questions. You've already answered them throughout the session. Um, so
I think there's
there's just one question that has popped up at the last minute. The question is, we have seen very interesting, eye-catching genome annotation figures. Which is the best software slash server to make these?
uh, for me the one that I like more to generate this kind of image is iTools. So this is
iTools.
yeah, iTools. I can wrote
Yeah,
in the
if you can
chat.
type the link, I can send it to all the participants.
Yeah, iTool is yeah, it's quite. You need to learn a little bit about, but it's quite is the figures that you can get with that is are really amazing. Then if you are able to manage quite well with the with the air program, for example, or other command lines, is also there are many types of tools that you can use to improve the figures. But for people who has not lot of experience, I think that works very well.
Okay, all right. Thank you so much. Thank you, Lorena, for the excellent 3 hours that you have spent with us. And I will see all the participants next time for another session, wonderful session on similar lines about using genomic indices for microbial identification. So thank you so much. Thank you, everybody. Thank you, Lorena, and hope to see you next month. Bye.